So, dear ladies and gentlemen, thank you for joining our presentation here as this webinar. The situation in the world is as it is. We can't change it, so therefore we can unfortunately not meet physically, but now only digitally. Nevertheless, I am thanking you for taking the time to join us. My name is Jörg Hollidt. I have the pleasure to be the chairman of the board of the Diagnostic Net of Berlin-Brandenburg, and on behalf of the Diagnostic Net, would like to introduce you to an interesting topic and to our exposition here on the Analytica. The topic of today is to give you an overview of currently available SARS-CoV-2 antibody tests, and I briefly show you the topics of today. So that you can join the presentation without being disturbed by me, I will switch off my video presence here, my video camera. I'll see you on the end of this presentation again, but for the time being, please enjoy the presentation and not being disturbed by me. So the targets of today, the topics of today, we have different targets and different results coming from different test systems. What are the principles of evaluation and to screen for validation of test systems? First topic, then overview of available SARS-CoV-2 test systems. Antibody test is the topic. So not antigen, not PCR. We are just focusing here on antibody tests. The consideration concerning preciseness, so theoretical and experience-based reflection on specificity and sensitivity of antibody test systems, and also to close it up, new targets and combinations, the way leading to a reliable result. First of all, who is the target, or what is our so-called, so to say, who is the enemy? SARS-CoV-2. It's the pathogen that caused the global COVID-19 outbreak ending last year and still continuing and even growing in the moment, at least in Germany. It is an envelope positive single-stranded RNA virus. Therefore, it is more sensitive to countermeasures than it would be. It would not be an enveloped virus. So therefore, that is a good position for us to fight the virus. It belongs to the family of coronaviruses for their crown-like appearance on the electron microscope. This is why it is named like this. And we will come to the family of coronaviruses in a bit. You see a sketch of the virus down there. We have several interesting proteins. We will go into detail on them in a bit, but just to make one thing clear, in the moment the world is focused on the spike protein, which is enabling the virus to enter into the host cell. It is the viral receptor protein for adhesion to the host cell receptor, and the capsid protein, nuclear capsid protein, that is the protein code around the genome of the virus. So if we continue that a little bit, 2/3 of the 5-ending genome encodes polyproteins, which are cleaved into 16 non-structural proteins, forming the replication complex, and from the other side, the 3 of the genome that encodes for structural proteins. And structural proteins are the one that are of interest for the diagnostics, diagnostical approaches. It's the spike protein, the envelope and the matrix membrane protein, and the nuclear capsid. And especially the spike protein has some peculiarities. You see that on the sketch on the right side. It is cleaved into an S1 and S2 subunit, and directly on the hinge where they still belong together, there you will find the receptor binding domain that enables the binding on one side to the TMPRSS2 as well as to the ACE2 receptor. The binding to both of these receptors on the cell surface, so here in light blue, given as the cell surface. The binding to both of those is mandatory for the virus to enter into the cell. And more general, the TMPRSS2 mechanism, this is a very general mechanism, and ACE2 is not specific here for the virus, or more specific. And so you see that this is obviously a good target to measure antibodies. If you have antibodies against this domain, the binding receptor binding domain or the spike protein, you are very likely to have protecting antibodies, neutralizing antibodies that hinder the virus to enter into the cell. And that is of importance. It's not of interest to know whether you have antibodies in general against the virus. The interesting question is, and that is giving you protection, is whether you have antibodies that are hindering the virus to enter to the cell, to the host cell, or whether you have antibodies that are in one way or the other blocking the virus and therefore having a neutralizing, a protective character. Where are we with a variety of coronaviruses? What we know is, of course, that this is a zoonotic virus. It has some highly pathogenic relatives. We know very well SARS-CoV-1, so to say, from 2002. It has had about 9,000 infected people, of which 774 died. Very high mortality rate, and also MERS-CoV-2 even higher. We know worldwide around 2,500 infected people, and out of them 858 deaths. Both of those viruses are managed in the moment, so it is an obsolete disease for the time being. But of course, we've learned a lot coming from that diseases. There are a couple of low pathogenic common coronaviruses which are related to the SARS-CoV-2 that is bothering us in the moment. You see them here. Always it's a human coronavirus, and then you have OC43, HKU1, NL63, and 229E. All of them are of minor importance. They are not very pathogenic. They are mostly harmless, taking care about, you know, some common cold features in normal people. You survive them. There are they are not much of an annoyance. The prevalence rate on the other hand, is huge. So 40 to 80% of the public of the people are being infected or having had an infection with this virus, and therefore we have to take care about these viruses very carefully in terms of diagnosing. Cross-reactivity is here the keyword. We have to take care whether you have a cross-reactivity towards this virus, yes or no, and it is completely unknown until up to now whether having had these viruses and to have an antibody reaction against these non-pathogenic or low pathogenic viruses is helping you in managing the SARS-CoV-2 infection or the COVID-19 disease, or whether this is deciding whether you have a more mild disease or a very severe COVID-19 disease. So that is unclear to the moment. Both ways are possible, and it is definitely a target that we have to follow, and it's not clear yet. If we go for the similarity between the coronaviruses, what you see here is a comparison of the nucleocapsid protein of SARS-CoV-2 and SARS-CoV. And you see they have a 90.5 sequence identity, and the similarity between those is 97.2. So it's a huge similarity between them, and there you see what is the target for a good diagnostic tool. It has to differentiate between them very, very strict. That is okay. An autoantibody or a test system should be able to differentiate between them, but do all the people in the industry and in the market at the moment and in the laboratories, do they do this properly, yes or no? And that is obviously one of the questions that have to be asked. And also, if you look at this, The spike protein of SARS-CoV-2 shares a high amino acid sequence identity with other coronaviruses. So SARS-CoV-2 and SARS-CoV, again, to compare them, they have a 76% sequence identity, and the S2 subunit 88% sequence identity. So this is definitely a task where routine diagnostic has to differentiate very, very precisely. Therefore, sequence and conformational conservation suggests in the end a cross-reactivity of neutralizing antibodies. So there is good and bad in the same thing. Usually you assume that if you have a similarity, you've had one of those diseases, not SARS-CoV, but let's say one of the lower pathogenic coronaviruses, that the antibodies you build against those may help you in neutralizing and coping with the new infection with the SARS-CoV-2. And also another thing is glycosylation, here shown on the SARS-CoV-2 spike protein. And if you see that you have to know that glycosylation, of course, is influencing folding, viral pathogenesis, priming of the host proteases, and also the antibody recognition. And if you look at this, there are 22 glycosylation sites per polymer, and at least 17 of them are usually occupied. And if you look at the sketch, the light blue is the protein scaffold, and on the right side with the dark blue added, you see the glycosylation with the glycans here, and you see it's completely different look from the outside, and therefore you have to see whether the antibodies are going against the protein structure or the proteins and the glycosylation, and that has to be differentiated, of course. And the glycosylation is always depending on the actual situation of the patient, of the donor, or whatever, in terms of his metabolism. So it's a huge difference in the early onset of a disease and the late onset. There are differences in the degree of glycosylation. There are plenty of antigens for SARS-CoV-2 already commercially available. Several antigens for SARS-CoV-2 for nucleocapsid protein, for spike protein, full length as one subunit, terminal domain, and the like. And if you just go for the commercial platforms, you have Biosol, you have more than 280 hits, or Antibody Online, just to name two, for 241 hits, telling you there are a lot of antigens available against which you can measure antibodies, but which one to take and how to validate that is the task of the diagnostic industry, of anybody who's doing research or work on that. And another thing is the seroconversion and the immune status of a patient. So while we know that SARS-CoV-2 is different, in SARS-CoV-1 we had the IgG detectable for 4 to 5 months, and just decreasing slowly 2 to 3 years after seroconversion. And for MERS, for instance, we have neutralizing antibodies persisting at least up to 34 months after recovery, so having a long-term protection at least for some years here. We do not know yet about SARS-CoV-2 precisely, but it is clear already that we have a different situation. So in the graph, you see the onset or the beginning of the detectability of the virus via PCR, and that detectability is going down quite quickly. So if the patient has the full-blown symptoms, then usually the PCR is negative already, and you cannot detect this anymore. But then, and that is specific for SARS-CoV-2 or very peculiar here at that point, IgM and IgG are coming up quite on the same time, and IgM is going down quickly, and the more riped IgG with a higher specificity and more power behind that in terms of immunity is also trailing down. So we do not know how long the protection will last, whether reinfection is possible. That is all not clear yet, but you see these curves will have a huge influence on the sensitivity and specificity of this test, as will be shown in the next few graphs. Here you see just a selection of different manufacturers of these test kits. They offering a lot of different test systems. They are on the market. They are certain, to a certain extent, all of them are validated. We see the classical CE IVD certification to bring a test kit on the market, but also we see FDA, and FDA especially has here taken into account an emergency certification, so you can bring it's allowed for the manufacturer to bring the test kit on the market without going through the full means of registering a test kit. It is said that it is on the same validity, but just quicker. Nevertheless, some additional has to be taken into account, and you have to revalidate that as a user in routine diagnostics. And if you look at this is all being taken from the homepage of the FDA by themselves. But what we see here is you have the antibody classes that are being detected, IgG, IgM, IgA in different variations, and also you have the different quality of antigen, nucleocapsid, S1, S2 domain. Even the combination is possible in ectodomains and only the receptor binding domain as well. So all of these variations are possible. Just to go a little bit more into detail, and from this now known more than 400 authorized diagnostic test kits, 170 are authorized as a serological assay system on different platforms, therefore the difference in number. And all of these, if you take the total one, then 125 have a CE mark and approval to be sold in Europe. Only 47 have the emergency use authorization for the FDA, and the FDA tells about 134 not to be distributed. And if you go to the right side of the picture, you see two diagrams as well. From this 31 authorized serological assays from the slide before, you see that 7 of them measuring IgG, 14 measuring IgG and IgM, and another 11 total IgG, so the combination of all of these subclass of immunoglobulins. And on the total right of the diagrams, which antigen is being measured, and here you see the distribution between spike protein, nucleocapsid protein, or a combination of both of them. So all the variations are on the market and available, and the user has to decide for himself which to use and validate by itself. There's no final validation or final opinion on that on the market yet. Just to show some selected, and they're randomly selected just to show them of 4 manufacturers. The antigens being used in this test systems are the known variation S1, S2, so the S1 domain, S2 domain of the spike protein, the nucleocapsid protein, or a combination of all 3 of them. Test principles are usually varying, so you find everything that is possible on the market: magnetic beads coated with the protein, double antigen sandwich, clear format, or ELISA format. Everything is possible, and as we see it, as is very typical in measuring antibodies by themselves, you have for each and every test system you have a different cutoff, so you never can compare the result of them. You always have to relate them to the individual cutoff of the respective test system. Therefore, if you would like to compare the results, you have to reference to the cutoff value of the test system. So a 10 is not a 10 in the other test system. It's always relative to the cutoff. And you have you have a lot of variations how the cutoff is being given. Sensitivity is sounds good. It's huge. It's about 90%. That is not sufficient for screening, but for individual testing, this is quite okay. The specificity is also quite high for a routine laboratory to measure antibodies. This is our numbers we are used to and can work with in routine laboratories, but when you have to take into account that this is measured on blood donors, and the question, of course, is these blood donors are when are the samples taken from the blood donor? So before the outbreak of SARS-CoV-2 or after, and if it is after, we would expect a higher degree of showing up of positivity. So there are some questions to be asked, and the future will tell us where we are and how do we define the specificity under this background compared to before and after outbreak of the disease, the pandemic. Interferences, I do not want to go into detail with that, but you see, of course, that it has to be measured. Just to take the first line, the non-pathogenic or low pathogenic human coronaviruses have to be measured, and also the other highly strict or highly dangerous coronaviruses have to be differentiated, otherwise you have a big problem. To show your attention, or to draw your attention on this specific situation here, we have a variation of the sensitivity of a test system, and that is holding, of course, for all of them, but just this company is mentioned that explicitly. The sensitivity is depending on the time after onset of the disease and measuring the antibody. So it's going up from 40 to 100% in the course of time. And to expand on that a little bit more in detail, the dependence of sampling day is significant. Here it is given between, so we always start at T0 with the syndrome, the onset of syndromes to be taken as the onset of disease here, and the variation is between 0 days and larger than or more than 12 days, and the number of samples are given. But what you see is right on the right side, the sensitivity is increasing from 8 up to 100%, and that is obviously clear if you take into account the graph. I showed some transparencies before. It takes a while before the antibody production rises and the immune system kicks in. And so we always have to see at what time do we use that, and what is the conclusion of a positive measuring value or a negative result in an antibody test system. So positive and negative predictive value are obviously very much depending on the context, on the time, on the population you are measuring. So in conclusion, we have a diversity of viruses, and we have a very specific and peculiar immune response towards the virus. The clinical use has to be shown to a certain extent already, and the setting in which to use this routine test is definitely restricted in the moment to certain situations and settings. The clinical development of the disease has to be taken into account. As mentioned, onset of symptoms is not truly onset of infection. It's not truly the idea when can you measure the virus on PCR level, and later on you see the antibodies, and how long the antibodies are being measured is still unclear. Are they protective, neutralizing? Next question. We do not know yet. It seems to be a hint towards that, and also whether you have a mild or severe follow-up of the disease. The time after measuring, harmonization and standardization in test systems is absolutely a question, and for antibodies always tricky. Only if we can see that we have a neutralizing antibody, we have a chance to say whether a patient is protected or not, and therefore the predictive value is to be measured and given, taking all this into account. It's not as easy as in other diagnostics here. We have to learn more about the virus. So we are on an exhibition, we are on Analytica 2020. We are here virtually with you, just digitally, but who do we have as our members joining the exposition here and presenting to you their capabilities? I go briefly through all of them, starting with the Invent Diagnostica. Here you see a lot of products and services for the research of SARS-CoV-2, especially is the focus on human samples, human biomaterial. The standard products offered comprise panels of a variety of patients directly positive in SARS-CoV-2, or the diseases to be differentiated, bulk material, large volume pools and controls in different matrices and volumes, positive and negative samples, as well as material to differentiate related diagnoses. and parameters, and also parameters of general interference. And this can be customized for your R&D project and of course being used in routine diagnostics. So human biomaterial for better diagnostics. ASCA is a company having a high potential in the production of monoclonal antibodies and recombinant proteins. Both of them are essential to build diagnostics in this field. And the advantage here is there's a small scale facility for research and target discovery, but that can be taken over to an upscaling large scale of production larger than 5 grams, and that is already quite a number if you consider this to be year source for routine production of diagnostics. And of course, this can all be done individually. It is very flexible and independent, and contract manufacturing for your protein needs. The expertise goes way back 30 years. Engine, the biomarker company, this is a platform to identify new targets that are relevant in every disease. Here, of course, SARS-CoV-2, it's basically a protein array comprising more than 30,000 proteins being printed on a matrix as a protein array. You can measure antibodies and autoantibodies against nerve cells, for instance, SARS-CoV-2 mediated autoimmune processes. And we know that more than 40% of people surviving COVID-19 suffering in the aftermath of severe neurological diseases and have to be treated heavily on that. So Guillain-Barré syndrome being caused by an overreacting immune system is one of the examples. And all of this can be investigated here, and also, of course, to differentiate between clusters of mild diseases and more severe cases. This is a good tool to investigating that is matrix independent. You can use serum, cerebrospinal fluid, cell supernatant, and the screening services available. So in the end, we do not know enough about SARS-CoV-2, and this may help us to enlighten that. Seramoon is a trusted company since long for the manufacturing, providing, and distributing of ELISA and microarrays to optimize here specifically the SARS-CoV-2 antibody diagnostics. There is a test for the receptor binding domain tested with SARS-CoV-19 positive and negative patients available. Sensitivity for this test system is high. I explained how to interpret these numbers a little bit earlier. The SeroSpot microarrays has the variety of different antigens here, even 4, and then antibody profiling is possible for SARS-CoV-2 infections. So that is a specific detection of SARS-CoV-2 antibodies on a routine level already. The University of Potsdam, one of our members, especially the professorship for immune technology, is able to implement innovative technologies for antibody production. This is more fast and more efficient, so very useful in rapid progress for R&D projects. The combination of monoclonal and recombinant antibody production is possible, and the generation of specific antibodies from human, murine, and cameloids origin is possible. So this is a specific set of antibodies, very useful to establish routine diagnostics under certain circumstances, and the assay systems can be pre-developed with the University of Potsdam. ICS, clinical services. On the next steps, you have a routine test system or test systems. You're asking yourself, is that working? You want to bring your own test system into the market, get the approval according to the in vitro diagnostic directive or regulation of the EU. You can do this with their services. The analytical performance, clinical performance can be measured in clinical studies, to plan them, to conduct them, to file the report, and the capacity to generate and get and procure the necessary biosamples of human beings, very specific procurement, and that is ethical, proper, and also scalable. And the recruitment of patients and donors is within their hand. So if you want to have a CE label on your routine test kit, or you want to validate your research approach, this is the address to go to. And then more basic, if you would like to detect whatever necessary in terms to fight the virus, has to be detected very precisely and in very low concentrations. Spec from the University of Potsdam as well is your partner for inline process analytics, photon density wave spectroscopy, and also applied analytical photonics for quality control, process safety, or resource efficiency. All this you can measure and steer your process and control your process is available here at the University of Potsdam. The next partner would be Central Biohub, where you can buy those already discussed biospecimen online. It's a marketplace for human biomaterial, and to our knowledge, it has the largest selection of SARS-CoV-2 and COVID-19 samples, serum, plasma, swabs, whatever it takes. A large variety of suppliers, so a lot of companies, or the companies that are on the market, present their samples here, so you can have what you see is what you get. You have a full transparency about specifications, suppliers, and prices. You can compare the clinical parameters and the sample information, and the samples are available immediately and will be shipped within days. So it is like a marketplace. It is what you see is what you get. So for immediate question, this is the immediate response. Another partner is the Zifin Diagnostic Berlin-Brandenburg. So they produce monoclonal antibodies as a contract manufacturer for IVD, large scale, very high expertise. And also they produce the dehydrated culture media for microbiology, products and recipe optimization according to your requirements for everything that goes left and right towards routine diagnostics. And last but not least, we do have the Cybertron, your partner for lab automation or automation of your processes. Long expertise in the field, ISO certifications are given, and they are compliant towards that. So any process you would like to automate and steer and to bring on the next level of reproducibility, Cybertron is your partner. Please go and visit their booth here on our virtual exhibition. For today, I would like to thank you for your attention. I will switch on, going back. Switch on the video again so that you can see me. So I thank you very much. It has been a pleasure to not see you today, unfortunately, but hopefully we will meet. I hope you find this interesting. If you have any further question, what you can see here is to the left, that is me. I had the pleasure to be a speaker of today. For further inquiries about the Diagnostic Network and about our partners, either visit them directly or get in touch with Hannes Thornagel, who is responsible to answer your questions, help you out, and guide you through the really impressive capacities and capabilities of the Diagnostic Net of Berlin-Brandenburg. So for today, stay healthy, stay with us, and looking forward to getting in touch with you one way or the other. Goodbye for today.